Good afternoon, students. Yesterday we have discussed about the innate immune system elaborately, where we have discussed about the different types of physiological barriers, anatomical barriers, and other cells. And there are some proteins which you are finding in the complement system. There are some proteins you can find in the innate immune system, that is complement proteins. These complement proteins will circulate in the blood serum as in inactive form. The complement system is useful for the clear the immune complexes or antigen antibody complex which is found in the blood serum. Today we will discuss about the what is complement system, what are the different types of complement proteins, and what are the different types of pathways which we are finding in the complement system, like a classical pathway, alternative pathway, and lectin and mannose pathway. These are the three pathways which we will discuss in the complement system. Maximum we will try to discuss today about elaborately about the classical pathway. Okay. Then coming to the first slide of the complement system, that is history of the complement system. In history of complement system, yes, the complement system mainly, the concept of complement system began in 1890s when Julius Bordet at the Institute of Pasteur in Paris, he started about the research about the complement system concept. And what he has done, he has taken the anti-serum of the sheep to the bacteria, against the bacteria of the Vibrio cholera. And when he has taken the anti-serum of the sheep and bacterial culture of the cholera, then he observed the lysis. Then he observed the lysis of the bacteria when he has added the anti-serum to the sample. Then, once again, what he has done, he heated the anti-serum which he has isolated from the sheep, then added the bacterial culture. Then he didn't observe the, what is that, same lysis. That means the anti-serum is having some of the protective proteins or the causative agents which can causing the or which can lysing the bacteria. I mean to say that sheep anti-serum is having some protective proteins. That protective proteins are useful for the lysis of bacteria culture or bacterium. When you are heating this anti-serum, the proteins which is present in the this anti-serum is degrading or denaturing. When you are added this heated or denatured proteins anti-serum to the bacterial culture, it is not harming the bacteria. I mean to say that. That means what it has given a conclusion, the anti-serum is having the some of the protective proteins which is causing the or which is killing the or which is lysing the bacterial culture. Then he named that substances which is present in the anti-serum which is protecting against the bacteria as alexins. That means the concept of the concept of complement system began with very simple experiment. He has taken the bacterial culture, he has added some anti-serum, then the proteins or the substances which is present in anti-serum is causing the lysis of this bacterial culture. Then same thing, what he has done, he has taken the heated anti-serum which is causing the or which is killing the bacteria, which is not killing the bacteria. Then he said that some of the substances which is present in the anti-serum causing the bacterial culture. Then he named the substances as alexins. But he doesn't coin the name as complement proteins. He doesn't coin the name as complement proteins. But who coined the name complement proteins as complement proteins? Because Paul Ehrlich coined the name or the term as complement. Why he has given or why he has coined this name, the substances too as complement? Because these complement proteins are complementing, are helping the main immune system to destruct or lysis the bacterial culture. Lysis the bacterial culture. You please, bacterial culture. Then after giving the 1890s, then the main concept of complement proteins as enhanced like anything. It is named as complement system because it was first identified as heat-labile component of serum that complemented or augmented the antibodies in killing the bacteria. Just now I said that the main immune system can kill the bacteria by releasing the different types of T cells, by releasing the different types of antibodies. I mean to say that the antibodies are attaching to the immune complexes, antigens, then forming the immune complexes. These immune complexes are cleared by complement proteins. Are these immune complexes cleared by the immune system when the complement proteins are sending the signal to the main immune system by different pathways? Like this, that's why the name came as complement. You see that the name here, complemented or augmented. Augmented means antibodies try to kill the bacteria, or try to clear the bacteria, or try to kill the antigens. This is augmented by or supported by some of the serum proteins which is present in the anti-serum. That's why Paul Ehrlich has coined these serum proteins as complement proteins. The complement proteins the complement system consists of serum, cell surface proteins involved in the defense against the pathogens, and tissue damage mediated by antibodies. The complement system is a major effector of cellular and humoral branch of immune system. Just now I told that the complement proteins are mainly belongs to the innate immune system. Some extent you can also add these complement proteins as adaptive or acquired immune system when the complement proteins are sending the signal to the main immune system by small subunits. But mainly the complement proteins are serum proteins. They are mainly comes under innate immunity. And as I said that this complement proteins mainly supporting the main immune system like cellular and humoral, like T cells and B cells. That's why it's called the major effector cellular and humoral branch of immune system. Plays a major role in both innate and adaptive immunity. That means complement proteins plays a major role in both innate and adaptive immunity. Complement system represents a group of proteins. The number will be like 20 to 30 proteins, serum proteins, which complement the immune system by clearing the antigen antibody complex. This words explaining that the complement proteins, the number will be like 20 to 30 proteins. You can say that especially in classical pathway, classical pathway, alternative pathway, and lectin and mannose pathway, lectin pathway, you will find the complement protein from C1, C2, C3, C4, C5, C6, C7, C7, C8, C9. Like C5, C6, C7, C8, C9, they will make a membrane attacking complex and attach to the cell membrane of the bacteria and lyse the cell membrane by releasing the different types of lytic enzymes, granulozymes, like other lytic enzymes. Then most of these proteins are found in the serum or on cell surfaces. Just now I said that. Most of these proteins mainly found in the serum, blood serum, as well as other cells like macrophages, phagocytic cells. They looks like membrane proteins. But what is the main organ or source of this complement protein synthesis? The main synthesis or main source of complement protein synthesis is liver. That means liver will synthesize the complement proteins mainly. Some extent, you can also say that gastrointestinal tract, you can also say that macrophages can synthesize or urinogenital system can synthesize the complement proteins. But if you want to say complement proteins are mainly synthesized from the liver. As you can see here, synthesized in liver as innate precursor and activated by the proteolysis during their interaction in sequential manner. Also produced by the blood monocytes, tissue macrophages, and epithelial cells of the gastrointestinal tract and urinogenital tract. I mean, the slide will explain you complement proteins are more or less 20 to 30 proteins, like C1 to C9, like C30, like C20, like that. But the main proteins from C1 to C9, which they are involving in the classical, alternative, and lectin pathway. Most of these proteins are found in the serum of the blood, as well as cell membranes like phagocytic cells, epithelial cells, and tissue macrophages. Then it is synthesized mainly by the liver, but to some extent it is also synthesized by the, what is that? Blood monocytes, tissue macrophages, epithelial cells, which is present or which is covering the gastrointestinal tract and urinogenital tract. That means, keep it in your brain, mainly the liver will synthesize the complement proteins. Some extent, epithelial cells of the gastrointestinal tract, urinogenital tract, blood monocytes, tissue macrophages can synthesize this complement proteins. Next, what are the general properties of these complement proteins? Just now I said that they are present in the serum of all animals, but its concentration is maximum in serum of guinea pig. Complement of one of the species are able to retract or react with the antibodies of the other species, but not the same extent. C proteins constitute about 5% of the normal serum protein. They are glycoproteins, are synthesized rapidly in inflammatory responses, hence are called as acute phase proteins. Heat liable and last activity at 56 degrees centigrade if you heat at 30 minutes. Immunoglobulins are not inactivated at this temperature. Then binds with the FC regions of the immunoglobulins. That means the general properties of this complement proteins are mainly these complement proteins. You can say that they will go and bind to the antibodies, FC receptors. If you see the immunoglobulin structure, the immunoglobulin is having the two main structures or two main parts, FAB region and FC region. FAB region means antigen binding site which is present on the immunoglobulin, that is called as FAB region. FC region means the FC region of the immunoglobulin attached to different types of immune cells. At the same time, this FC region is, if it is attaching to the antigen, this FC region can activate the complement proteins. Then this all complement proteins mainly present in the blood serum. Then all complement proteins belongs to glycoproteins. Mainly the rapid synthesis of the complement proteins at the time of inflammation. That means at the time of inflammation means we already discussed in the yesterday's class what is inflammation. Inflammation is an immune response against the infection. When there is a damaged tissue or a damaged organ or a damaged cell, it has to clear from the particular organ or particular area or in the body. But how it will be clear? When the immune response is more in view of inflammation, then complement proteins will be activated. Then complement proteins will come and bind to that activated area and clear the bacteria or pathogen by activating different types of pathways. Then the complement proteins are, we already discussed it in the first slide, they are heat liable, but antibodies are not heat liable. Some extent, if you beyond like what is that 60 or beyond like what is that 80 up to or 1 hour, 2 hours, then this protein is also denature. But complement proteins can denature at 50 degrees centigrade if you heat up to 30 minutes. Then the complement proteins will be denatured. That's why the denature means it will lose its activity. That means if you heat the complement proteins at 50 degrees centigrade for 30 minutes, then they will lose its activity. That's why we are calling the name component proteins are heat liable. That means heat sensitive. Then the component proteins are binding to the Fc regions of the immunoglobulin. That is very important. Don't think that the component proteins will go and bind to the FAB region. FAB region is attached with the antigen. Then there are three main effects of complement activation. When the complement proteins are activated, lysis of the cells, because sometimes the antibody will go and bind to the different types of molecules which is present on the membrane of the bacteria. Sometimes this, directly the antigen attached with the antibody, then forming the immune complexes. Sometimes if you are undergoing to the grafting, or if you are undergoing to the transformation of the different types of organs from other sources, then it will be detected by component proteins, then activate, then send the signal to the main immune system. We have detected a foreign particle or a foreign organ, then our bacteria, then send the signal and also activate the classical alternative and lectin pathway and make the membrane attacking complex and lyse the cells. This process we are calling as lysis of cells, mainly bacteria, allografts, and tumor cells. Generation of mediators of inflammation. Just now I said that in inflammatory responses, the main concentration of the component proteins will be increased or enhanced like anything. But when? But how? Mainly, when component proteins are activating, the split products will be formed. Smaller subunit and larger subunit. This smaller subunit will work as an anaphylaxis factor. That means that you can also call it as A subunit. This smaller or A subunit will send the signal to the main immune system. We found a infected organ. We found a foreign pathogen. We found a new organ which is not related to us. In the main immune system will recruit the different types of immune cells at the area of infection, then clear the pathogen. That means generation of mediators of inflammation. When the inflammation will start, there is a recruitment of high number of immune cells like natural killer cells, phagocytic cells, macrophages, by releasing the cytokinins, by releasing the interferons. It means they will release the chemical mediators. This chemical mediator will send the signal to the main immune system. The main immune system will recruit the different types of immune cells at the area of infection. This is called as generation of mediators of inflammation. Then the last one that is called as opsonization, enhancement of phagocytosis. As I said that component proteins are mainly synthesized by the liver, but to some extent phagocytic cells and tissue macrophages can also synthesize the component proteins. And component proteins are circulating in the serum in an inactivated form. And also some component proteins are membrane attached proteins. Mainly the phagocytic cells is having the component proteins are attached. When component proteins are activated, the phagocytic cells will be activated and engulf the or opsonize the bacterial cell, then cause the or kill the bacteria, a process that is called as phagocytosis or opsonization. This is called as opsonization. There is a process in the opsonization that is called as, in the immunology, that is called as opsonization. Especially macrophages and phagocytic cells, they will go and form a lobopodia-like structures around the pathogen, then form a small vacuole, then intake the bacteria into their cytoplasm. Then release the different types of lytic enzyme around this vacuole and lyse the cell. When their lobopodia is forming around this pathogen, think that this is a pathogen. This pathogen is covered like this by the lobopodia-like structure, pseudopodia-like structure. Now this pseudopodia will form a vacuole-like structure. This is called as opsonin. This process is called as opsonization. And after opsonization, this will intake the cell pathogen into the cell body and release the different types of lytic enzymes and digest the pathogen. This process is called as opsonization. This opsonization, inflammation, and lysis process mainly come to the body by activation of complement system. We discussed that over 30 serum and cell surface proteins are present in the complement system. And complement components are designated as by number. Components, we said that 30 proteins are there. Out of these 30 proteins, we will give we will coin the number like C1, C2, C3, C4, C5, C6, C7, C8, C9. Then you can also give, if you find that alternative pathway, you won't find that like C1, C2, C3, C4, C6, C7, and you may also find that factor D, propyridine, lecithin like that. It means in complement system, you may find the numbers like C1 to C9. You may find the letters like factor D, propyridine. In serum, inactive, activated sequentially as a cascade. That's why you can also say that complement activation process is a cascade pathway. Complement activation is a cascade pathway means if you activate one protein in the complement system, if you activate one protein or first protein in the complement system, it will take care of the activation of all other proteins. No need to activate other proteins directly. If you activate C1, it will activate the C4. If you activate C4, it can activate the C2. If you activate C2, it will activate the C3. If you activate C3, it will activate the C5, C6, C7, C8, C9. I mean to say that if you activate one protein, it will take care of the activation of other proteins which is present in the complement system. Such a type of method of activation we are calling as cascade mechanism. Cascade mechanism. Then that's why the pathways which we are finding in the complement system, we can say that cascade pathways. This is called as a meaning of cascade. Then complement receptors. Cell surface receptor recognize activated components. I already told that. Complement proteins directly detect the membrane-bound antigens or immune complexes. It is also activated by other complement protein split products. For example, if you take the C2 or C3, C3 can activate, if you activate it, it will split into C3A and C3B. Then C3B can activate the other protein. But C3A cannot activate because this smaller subunit will work as a chemical mediator to send the signal to the main immune system. That's why they are cell surface receptors recognize the activated components. Then regulatory proteins of the complement system. Both in serum and cell surface inhibit inactivated components. Then what is activation of complement system? That is very important. What is the concept of activation of complement system? Mainly, the activation of complement proteins means it will split into two subunits. It will split into two subunits. What are the two subunits? When it is in active form, it is a single subunit. When it is combined with the or hitted with the antigen antibody complex or other split product of the compound, are you able to hear my voice clearly, students?
Yes, sir.
Okay. Because somebody is telling that sir, the headset is not have. Okay. I got the doubt. Okay, no problem. If you are hearing clearly, it's no problem. Thank you. What is the meaning of activation of complement proteins mainly? Activation of complement proteins means you have to split the one complement protein into two small subunits. The subunits ratio also different. One subunit will be big, one subunit will be small. You can see here, complement proteins are pro-enzymes, activation by cleavage. That means by split. Take an example as a C4. If you are activating the C4 will be cleaved into or split into two subunits, that is the C4A and C4B. The larger subunit of the C4 is we are calling as C4B. That means keep it in your mind very precisely. Larger subunit of the complement protein is called as B subunit. Smaller subunit of the complement proteins we are calling as A subunit, but it is having a exception. Each theorem has have its own exceptions. Like complement proteins also having exceptions, except to C2. The theory, the rule which I am telling, that is right. That means activation of complement proteins means you have to split into two small subunits. That is called as activation. The larger subunit you can call it as a C4B. Smaller subunit you can call it as C4A or A subunit. But except in the C2, larger subunit will be A, smaller subunit will be B. You can see here. C4 is activated or split into C4A and C4B. C4B is a larger subunit. C4A is a smaller subunit. A is a smaller fragment or diffusion fragment. B is a larger fragment, remains binds to microbe and activate the other coming complement protein. That means the C4B will go and activate the C2. Then C2, coming to the C2, C2 have only one exception. The C2A is a larger fragment, C2B is a smaller fragment. That means this is the exception which you are finding in the, what is that, complement system. Then what are the different types of pathways which you are finding in the complement system? There are three pathways mainly you can find in the complement system, that is classical pathway, alternative pathway, and lectin pathway. You can also call it as lectin and Manus pathway. Classical, what is the meaning of classical pathway? Is antibody dependent pathway and triggered by the formation of soluble antigen antibody complex by binding the antibody to the antigen present on the target cell surface. I mean to say that to activate the classical pathway, antibody dependent pathway and triggered by the formation of soluble antigen antibody complex. That means the only one pathway which is dependent on the antibody to activate, that is called as classical pathway. You can see here it is an antibody dependent pathway and triggered by the formation of soluble antigen antibody complex by binding the antibody to the antigen present on its target cell surface. That means some of the antigens present on the membrane of the bacteria, these antigens attached with the immunoglobulins like this. Think that this is antigen, attacked by the immunoglobulin, the FAB region. Then this is called as FC region. This FC region can activate the complement systems. Alternative pathway means is an antibody independent pathway stimulated by the antigen directly, bacterial cell surface components. You see, this is the first pathway, that is called as antibody dependent pathway, but the second pathway is antibody independent pathway. Directly this pathway can go and attach with the antigen without in absence of antibody. The lectin pathway, it is also an important antibody independent pathway, but resembles the classical pathway. That means it is also independent, but it looks like in view of complement proteins, it is what is that? It is classic. What is it? Independent to antibody. I mean to say that the classical pathway, to activate the classical pathway, you need to have a antigen antibody complex. If there is no antibody, the classical pathway will not be activated. But in a different way, alternative pathway and lectin and Manus pathway, these two pathways can activate independently in absence of antibodies. That's why alternative pathway and lectin pathways are independent of the immunoglobulins. Independent of the immunoglobulins. Then stages of complement activation. How the complement activation will be there. You see the main goal of complement system, formation of C3 convertase. What is the meaning of C3 convertase? C3 convertase means C2A4B, that is called as a C3 convertase. Once this complex is forming, this C3 convertase can split the C3 protein and form the C3A and C3B. This C3B will go and bind to the C2A and C4B and forming the C5 convertase. Finally, it will form a membrane attacking complex MAC. In addition, and formation of C3 convertase are different in classical and alternative pathways. These then followed by the parallel route of activation process independently with the or without antibodies. That means if you see the stages of complement activation, first C3 convertase will form by forming the or by combining the C2A and 4B. Then once you form the C3 convertase, this C3 will take care about the C5. This C5 will as C5A and C5B. This C5B will go and bind to the C6, C7, C8, C9 and form the MAC, like what is the membrane attacking complex. This membrane attacking complex, like the structure is like a bore-like structure, which will go and bind on the cell membrane and bore the cell membrane, then lyse the cell by releasing different types of lytic enzymes. You see the initiation phases of the C3 convertase are different in classical pathway and alternative pathway. These then follow the parallel route to merge the C5 convertase stage R and finally generate the MAC by the common route. All these pathways lead to the production of C3B and the central molecule of the complement cascade. The presence of C3B on the surface of the microbe masks the foreign and targets of its destruction. C3B has two important functions. It combines with the other complement components to generate the C5 convertase and the enzyme that leads to the production of membrane attacking complex. And it opposes or opsonizes the bacteria because the phagocytes have receptors for C3B and on their surfaces. I mean to say that if you are activating the C3 protein, then C3 protein will take care of the activation of C5, then formation of the membrane attacking complex. Once membrane attacking complex is formed, it will take care about the activation of other complement proteins. Then some definitions we can find in the complement system, that is C activation, C fixation, C inactivation, convertase or esterase. What is the meaning of C activation? Alteration of C proteins or complement proteins such that they have to interact with the next component. C fixation means denaturation, usually by heat, of an early C component, resulting in the loss of hemolytic activity. Then coming to the convertase or esterase. Altered C protein which acts as proteolytic enzyme for another C component. As I said that complement proteins activation is a cascade pathway. That means if you activate the C1 protein, like if you take the C1 is having the three subunits C1Q, C1R and C1S. If you are activating the antigen antibody complex, if it is activating the C1Q, then it can take care of the activation of C1R and C1S. Once if you activate the C1R, then it will structurally modify the structure of the C4, then activate the C4A and C4B, then C2, then C5, then C6, C7, C8, C9. Like this, the complement protein activation process is a cascade pathway. Then what are the biological effects of the complement system? There are different types of biological effects to the component system. The first one is opsonization. Because if you see the type 4 hypersensitivity and type 3 hypersensitivity, some of the immune cells directly having the access with some complement protein related receptor like C3b and C1q and enhance the phagocytosis. That means if your bacteria is attached with the antigen antibody complex, this antigen antibody complex is attaching with the C3b or C1q, they can enhance the properties of the phagocytosis of the particular pathogen. Then chemotaxis. When the C5a is activated, it can also obviously activate the C5, C6 and C7 complex and attract the neutrophils, C5a, eosinophils, adhesive and neutrophils to the endothelium or epithelium. That means sometimes this C5, 6, 7 will works as a chemotactic factor. What is the meaning of chemotactic factor? Chemotactic factor means the factors can recruit the immune cells at the area of infection by releasing the chemical mediators. That is called as chemotactic process. Once again, I am telling you, the chemical mediators which we are using to activate the different types of immune cells like interferons, what is that, cytokinins, they're all chemotaxins. These chemical mediators are useful for the recruiting the different types of immune cells at the area of infection. Then you can call it as chemotaxis. Then the last one that is called as anaphylatoxin. As I said that the split products of the complement proteins, the larger subunit will be B subunit, smaller subunit will be A subunit. There is one exception with respect to the C2 protein. In C2 protein, larger subunit will be A, smaller subunit will be B. But what is the main role of smaller subunit? You can say that larger subunit will go and activate the other proteins, and also sometimes somewhere they will go and useful for the formation of the membrane attacking complex. But what is the main role of smaller subunit which is present in the complement system? The smaller subunit of the complement system like C3a, C4a and C5a cause the degradation of the mast cell, bind directly to the smooth muscles of the bronchioles and bronchospasm. This is about the anaphylatoxin. What is the meaning of anaphylatoxin? The smaller subunits works as anaphylatoxins like C3a, C4a and C5a. This smaller subunit will go and diffuse into the blood system and activate the main immune cells. Then main immune cells will recruit the different types of immune cells at the area of infection. Then opsonization and phagocytosis. This is the main function of complement systems. Opsonization means you can see here, these are the complement proteins. These are the bacterium. This bacteria is attached with the complement proteins. Then they will come and present to the monoclonal antibodies. That means the C3b is a opsonin. Opsonins are molecules that bind both of the bacteria and phagocytosis or phagocytes. Opsonization increases the phagocytosis by 1000-fold. Then cytolysis. That means including to the opsonization, chemotaxis and anaphylatoxin. There is one more function to the complement protein that is called as cytolysis. How the complement proteins will lyse the cell? As I said that finally the complement split products like C5b, C6, C7, C8 and C9, they will make a bore-like structure. This bore-like structure will go and bind on the and sit on the bacterial cell membrane, then make a bore, then it will lyse the tissue of the cell membrane of the bacteria, then kill the bacteria by this process. This is called as a cytolysis directly. Then enhancement of the antibody production. Binding of C3b to the receptors on the cell surface, activated B cells enhance the antibody production. Then what are the different types of activation pathways with regard to the classical pathway, alternative pathway, and membrane attacking lectin pathway? Antigen and antibody complexes. Length as a lectin bind to the pathogen surfaces. Alternative pathway means pathogenesis surface. All these pathways is present in the complement system is also work for the recruitment of inflammatory cells, opsonation of the pathogens, killing of pathogens. And you can see here the complement system can enhance its roots because initially the complement protein solely belongs to the innate immune system. But nowadays we are also discussing that the complement protein is in the acquired immune system. You see, the innate immune properties of the complement proteins like opsonization, lysis of pathogens, chemotaxis, inflammation, then disposal system, clearance of immune toxicity, and apoptotic cells. Then adaptive immunity. Under adaptive immunity, augmentation of the antibody response, production of T cell response, enhancement of self-reactive B cells, T and B cells. Then what is the complement pathway? I already told you complement pathways are 3 types. What are the 3 types? Classical pathway, that is the first and important pathway. Then you can also say that alternative pathway, and you can also say that what is that lectin and mannose pathway. What exactly you will find in the complement pathway? As I said that complement pathways are 3: classical, alternative, and lectin pathway. Under a classical pathway, complement is activated by antigen antibody complex. Complement is activated by antigen and antibody complex. Fc portion of the antibody form a binding site for C1q. That means the first protein which is activated in the complement system, that is the C1q. The Fc region, because as I said that the complement proteins will be activated by the antigen and antibody complex or immune complex. But where exactly this immune complexes will attach to the complement proteins to activate or to split? The Fc regions of the complement proteins get the conformation changes in the structure of the C1 proteins or C1 subunit of the, that is C1q, which is the subunit of the C1 protein. Then you can see here the numerical sequences of the complement factors, that is C1, C4, C2, C3, C5, C6, C7, C8, C9. That means the activation of complement proteins will be like this, because generally we will write like that C1, C2, C3, C4, C5, C6, C7, C8, C9. But the activation process of complement proteins will be like this. C1, if you activate C1, then C1 will go and activate the C4. Generally, what we will do, the C1 will activate the C2. No. C1 will activate the C4. C4 will activate the C2. C2 can activate the C3. C3 can activate the C5. C5 can activate the C6, C7, C8, C9. This is how the complement proteins serially can activate it. But why the C2 cannot be activated? Because the scientists who discovered these proteins, they have given the numbers. That means first C1 proteins will be discovered, then C2 has discovered. But C2 has not activated by the C1. That means the numbers or numericals has given to the complement proteins based upon the sequence of identity, not based upon the sequence of activation. Don't forget, this is very important. Then you see that in complement activation of the classical pathway, the FC region of the antigen antibody complex or antibody can activate the C1. The C1 can activate the C4. C4 can activate the C2. Then C2 can activate the C3, C5, C6, C7. That's why this pathway we are calling as classical pathway, which is dependent on the antibody. Then form a membrane attacking complex. You can see that. Then form a membrane attacking complex like this. It's just like a bore-like structure. Then the fluids of the outside the cell will enter into the cell like mainly granulozymes and other lytic enzymes. They can kill the bacteria or degrade the bacteria. That's why this is called as membrane damage. Then in classical pathway, the classical pathway is initiated by the antigen-antibody reaction. That's why this is which we are calling as antibody-dependent pathway. A specific reactive site on the constant portion of the antibody becomes uncovered or activated, and this in turn binds directly with the C1 molecule of the complement system, sitting into the motion of cascade sequential reactions. I mean to say that generally you will find the two types of regions like constant region and variable region. This constant region of the FC region can try to activate the C1 protein. Then generally the C1 in the form of a pro-enzyme, then when it is activated, it is converted as active enzyme. You see that beginning this with activation of this pro-enzyme C1 itself, the C1 enzymes that are formed then activate successfully, increasing quantities of enzymes in the later stages of the immune system, so that the form of small beginning and extremely large amplified reaction occurs. That means if you activate a single C1 protein, it will take care of a big pathway, activation of the big pathway. C1, C4, C2, C3, C4, C5, C6, C7, C8, C9, and form the membrane attacking complex and damage the cell. Proteins in the classical pathway. C1 exists in the blood serum as a molecular complex containing one molecule of C1q, two molecules of C1r, two molecules of C1s. That means if you see the C1 structure, if you see the C1 protein complement protein structure, it's just like a T-shaped structure. What is it? You can say that inverted Y-shaped structure. The arms of two Ys having the more or less 4 molecules of subunits like C1r and C1s, and one molecule of C1q. Combining, we say that totally 5. The constant region of the antibody contains a binding site for the C1q. The constant region of the antibody having a binding site for the C1q, a single molecule of IgM is enough to initiate the pathway. Mainly the complement proteins can be activated by IgG and IgM immunoglobulins. Binding of C1q activates the C1r and C1s. You see, this is the Q subunit of the C1 proteins. If you activate the, you see this is IgG antibody activating the C1q. FC region of the IgG antibody activating, and then is going and binding to the C1q. When you are activating the C1q, there is a structural conformational change in the R and S subunits of the C1 protein. Then activated C1 is cleaves to serum proteins. C4 is cleaved into a large fragment that is called C4b, which binds covalent to the sugar residues on cell surface glycoproteins. And a smaller and inactive fragments like C4a, which diffuses away. Then C2 is cleaved into C2b, which binds non-covalently to a site on the C4b, leaving a smaller inactive fragment as a C2a with diffuse away. The complex of C2b, C4b, and 2b is called as a C3 convertase. Now you see, that means if you activate the C1 protein, then it can activate the C4. The larger subunit of the C4 can activate the C2. In the larger subunit of the C2, that is called as a 2B, it's come and bind to the larger subunit of the C4, that is called as 4B, and forming a complex that is called as, you can see here, C4B and 2B. You can write the line above. If there is a line, then you can say that it's the active form of the complement protein. Is called as, that means the complex of C4B and 2B is called as a C3 convertase. C3 convertase means this complex can split the C3 protein. Then when C3 protein is activated, C3 is most abundant protein in the complement system. When the C3 protein is activated, the C3b, which binds covalently to the glycoprotein scattered across the cell surface, macrophages and neutrophils have the receptors of C3b and can be bind to the C3b coated cell, particularly preparatory to the phagocytosis. This effect qualifies C3b as an opsonine. Then this C3b fragment released into the surrounding fluids can bind into the receptors on basophils and mast cells, triggering them to release their vasoactive components like histamine. Because of this role of these materials in anaphylaxis, C3A is called as anaphylatoxin. Then some of the C3b binds to the molecules of C5, expose them to cleavage the C4b, 2b, and 3A. C4b, 2b, and 3A works as a C5 convertase. This C5 convertase will work as activate the other component proteins like C6, C7, C8, and C9. C6, C7, C8, and C9. I mean to say that when C6, C7, C8, C9 make a combined structure that is called as membrane attacking complex. This is the main goal of all complement pathways. This membrane attacking complex will go and bind to the cell membrane of the bacterial cell or infected area and lyse the cell by making the pores, that is holes on the cell membrane, and release the different types of granulozymes and other lytic enzymes to kill the bacterial cell. You can see here in the classical pathway, antigen antibody complex are activated in the C1q. C1 will be 1q subunit and 2 R subunit and 2 S subunits. There is a very, very important question you might get in the computer exams. Which of the following classical pathway component proteins can activate first or interact first by antigen antibody complex, that is the C1q. If you activate C1q, then there is a conformational change in the C1q. Obviously, it can change the structure of the R2, C1R, and C1S. That means the total conformation of the C1 protein will be changed. Then you can see here activating the C1q RS2 will be activated. This activated C1 protein can activate the C4, then it will give a smaller subunit and larger subunit. Larger subunit we are calling as the C4b, smaller subunit which we are calling as the C4a. Then this smaller subunit C4b and the larger subunit of the C2 called as C2a works as C3 convertase. Now this C3 convertase can convert or can split the C3 protein as C3a and C3a and C3b. Please change here. This is C3b. The C3a works as an anaphylactic factor, and the smaller subunit will go and bind to the different types of macrophages or immune cells and activate other immune cells and recruit the immune cell at the area of infection to clear the immune complexes. Then you can see here, now this is C3b will go and make the complexes. You see here, there is no line. If there is a line, R2 or S2 is activated. That means if you are drawing a line on the head of this some protein, then you can say that the protein is activated. I mean to say that you can see here the C4b doesn't have any line, but the C4b and 2a make a complex. This complex C4b and 2a complex is working as a C3 convertase. Now this C3 convertase is coming and binding or splitting the what is that? The next protein that is called as a C3. Now after splitting of the C3 as a smaller subunit and larger subunit, this larger subunit will go and make the complex with this C3 convertase like C4b, 2a and 3b. That means 4b subunit, that means the larger subunit of the C4 protein, larger subunit of the C2 protein, larger subunit of the C3 protein. Will make a complex. You see, this is line means complex, activated form. Now this activated complex will work as a C5 convertase. That means this complex will work as an enzyme, and this enzyme name we are calling as a C5 convertase. Now this C5 convertase can able to activate the C5 protein. Can able to activate the C5 protein. Now this is C5 protein in presence of C5 convertase split into C5b and C5a. Once C5b is formed in the classical pathway, obviously it's just like a cascade. It will go and activate the C6, C7, C8, C9 and form a membrane attacking complex. You can see here, this is the membrane attacking complex. This is called as membrane attacking complex. This membrane attacking complex will come and bind on the bacterial cell membrane and lyse the cell, making the holes. That means today we have discussed about the classical pathway of the complement system. I will give the gist of this today's topic. Complement proteins mainly belongs to innate immunity, but some extent you can also include in the adaptive immunity. And when you are coming to the immune responses, complement proteins mainly involved in the humoral immunity as well as cell-mediated immunity. Complement proteins are serum proteins. In the serum, they will be in the form of inactivated form. That is called as pro-enzymes. When they are encountering with the other complement proteins, or when they are encountering with the antigen and antibody complexes, then the complement proteins will be activated. Then there are more or less in the human body, 20 to 30 complement proteins are there. First time the concept of complement proteins is explained by Bardet. Then the name he has given as alexins. But the name complement is given by the Paul Ehrlich. The complement system augment the clearing of the antigens, especially it is used to antibodies or immunoglobulins to clear the process in the process of clearing the antigens or pathogens. There are three types of pathways which you can find in the complement system: classical pathway, alternative pathway, and lectin and mannose pathway. Why the classical pathway which we are calling as classical pathway? Because the activation process which you are finding in the classical pathway is just like classical. That means this is antigen antibody dependent. If you activate the C1, C1 will take care of the activation of C4. If you activate the C4, C4 will take care of the activation of C2. If you activate the C2, then C2 will take care of the activation of C3. Like this, you have a pathway. That's why this pathway we are calling as classical pathway. But what are the main important functions or biological functions to the complement system? As we have discussed in the previous slides, complement system are useful to clear the different types of pathogens by lysis, cellular lysis, augmentation to the antibodies, and other process like immune responses, releasing the chemical mediators like that. Finally, the main characteristic or the main goal of this all complement systems, they have to form the membrane attacking complex. Now, this membrane attacking complex will go and bind, sit on this cell membranes of the bacteria or infected cell and make the pores on this one, then kill that infected cell or bacteria. Then coming into the, what is that? Which portion of the immunoglobulin can activate the C1 protein? The Fc portion of the immunoglobulin can activate the C1 protein. If you activate C1, then C1q, then C1q will conformation change. Then this C1q will go and change the structure of the C1R and C1s. Now the C1s can activate the C4. Now this C4 split into smaller subunit and larger subunit. The smaller subunit of the C1 protein we are calling as C4A. The larger subunit of the C4 protein we are calling as C4B. This larger subunit which we are calling as the C4B will go and activate the other or coming component protein in that particular pathway. But what is the main function of this smaller subunit? The smaller subunit works as an anaphylactic factor. They will go and send the signal to the main immune system. We found or we attached a infected cell or pathogen. You please act against that one and recruit the different types of immune cells in the area of infection to clear the infected cell or pathogen. Now this C4B, you can see here, this is the FAB region where antigen is binding. This is the FC region. To this FC region, the immunoglobulin or the component proteins will be attached. Once the component proteins are activated, like C3, like what is that, C4, C4B will go and activate the C2. I already told the exception. The larger subunit of the C2 is A, smaller subunit of the C2 is B. The larger subunit of the C2A will go and make the complex with the C4B and form the C4B and 2A. Combined, you can also call it as a C3 convertase. Now, this C3 convertase can activate the C3. Now, the C3 will be split into C3A and C3B. The larger subunit of the C3B will go and complex with the C4B 2A and form the C4B 2A and 3B and works as a C5 convertase. Now, this C5 convertase can activate the or split the C5 protein. And the C5 protein is after activation, it is forming the C5A and C5B. And this C5B will go and activate the C6, C7, C8, and C9 and make a bore-like structure. You can see here. You see, this is a bore-like structure which will go and bind on the cell membrane and release the different types of lytic enzymes and kill the cells. This is how the complement proteins will make the membrane attacking complex in the classical pathway. Okay. Then you please read the basic characteristics of the complement system. You can see here the complement system, as I said, that it only belongs to innate immunity. But some extent is augmenting the inhibitors. That's why they kept in the adaptive immunity. And this is about the classical pathway. You can see classical pathway, what is the lectin pathway and alternative pathway. Final source is recruiting inflammatory cells, opsonation of pathogenesis, and killing the pathogens. This is very important diagram where all the pathways will come to the same goal, which is forming the membrane attacking complexes. Okay, thank you very much. If you have any doubt, you can feel free to ask. Any doubts? Hello? Sweta, Ramesh, Ravi Kumar, Seema Taslim. If you have any doubt, you can ask with regard to the classical pathway. If you don't have any doubt, then I can leave the class. Tomorrow we'll discuss about the alternative pathway and lectin pathway. If time permits, we'll also discuss about the immunoglobulin structure. Thank you very much.
Thank you, sir.
Thank you.